Cellular respiration and fermentation. First, let's look at cellular respiration. Cellular respiration is the process which breaks down sugar to the form of energy that they can use in the cell. This picture shows the overall process of cellular respiration. ATP, or known as adenosine triphosphate, is the energy that I talked about. It is the main energy of the cell. When ATP is used already, it's called ADP. While our body does cellular respiration, our body gets oxygen and removes carbon dioxide.
In cellular respiration, there are four main steps. First step is glycolysis. This step happens in cytoplasm, like here. Next, the second step is preparatory reaction. This step happens in matrix of mitochondria, like here. Third step is citric acid cycle, also known as Krebs cycle. This step happens in the matrix of mitochondria. The last step is electron transport chain. The final step happens in the crystal membrane of mitochondria.
So in cellular respiration, there are two types of it, which are aerobic respiration and anaerobic respiration. Aerobic respiration is the normal one that we usually use function. It requires oxygen, and anaerobic respiration is the special one that doesn't require oxygen. It only has one stage, which is glycolysis.
The similarity and the differences between aerobic respiration and anaerobic respiration. For the similarity, they are both normal parts performed in cellular respiration, and both of them can occur in eukaryotic organisms. Eukaryotes are organisms whose cells contain a nucleus and other membrane-bound organelles, including animals and plant cells. And for the differences, if oxygen is present, then the cellular respiration will do aerobic. If oxygen is absent, cellular respiration will do anaerobic. When we exercising, our cells do anaerobic respiration. That's also the reason why we have cramp in the day after. And some other organisms such as yeast, bacteria, and archaea do anaerobic respiration instead of aerobic respiration. However, many other organisms can perform either aerobic and anaerobic respiration, depending on whether oxygen is present. Cellular respiration is a catabolic reaction, which the breaking down of the bond between molecules and releasing the energy is present during the processes.
Glycolysis. Glycolysis occurs in cytoplasm of the cell, which is a liquid part of the cell. It is a metabolic pathway. Metabolic pathway is a linked series of chemical reactions occurring in a cell. The product of the previous reaction will become a substrate from the next reaction. Glycolysis involves 10 enzymatic reactions. It divides into two main steps, which are energy investment phase and energy harvesting phase.
Energy investment phase. Invest means to put into. In this phase, 2 ATP is used as activation energy to rearrange by attached phosphate from it, one from each ATP, causing glucose unstable and split into 3 carbon compounds with phosphates attached.
Energy harvesting phase. This is the second stage of the glycolysis. Once the 2 carbon compound lose electron or oxidize and become another 3 carbon compounds, NAD+, are reduced and become NADH. Then we got 2 NADH, one for each NAD+. These 2 NADH will be used in the electron transport chain, so it will go to the mitochondria, where the electron transport chain takes place.
As the 2 NAD+ come to carry the electron, they also bring the phosphate with them. Each of the NAD+ give one phosphate to each of the 3 carbon compounds, resulting 2 phosphates attached to each of the 3 carbon compounds. Next, 4 ATP is produced by donating 4 phosphates that attach to the 2 3-carbon compound to form 4 ATP. The 2 3-carbon compound will rearrange themselves into a different 2 3-carbon compound called pyruvates. As a result, 2 pyruvates, each with 3 carbon, are the final product. ATP is produced during this process. Within the process, 2 ATP is used in the energy investment phase, so 4 minus 2 equals to 2. so 2 ATP net gain in glycolysis. These 2 ATP is ready to be used by the cell for any cell processes. And for the NADH, 2 NADH gain in glycolysis, both of them will go directly to the electron transport chain to help make more ATP.
Input and output in glycolysis. Input are glucose, 2 NAD+, 2 ADP, 4 ADP, and 4 phosphate. In output are 2 pyruvate, 2 NADH, 4 ADP, and 2 ADP.
Next step of cellular respiration is preparatory reaction. Main idea for preparatory reaction is to convert 2 pyruvate to 2-acetyl-CoA. Preparatory reaction is the second step of cellular respiration that comes after glycolysis, and it is also called pyruvate oxidation.
Preparatory reaction happen in the matrix of mitochondria. Glycolysis that takes place in the cytoplasm produce 2 pyruvates. 2 pyruvates get into the mitochondria to begin the preparatory reaction.
Pyruvate is oxidized to 2-acetyl-CoA. When someone is oxidized, someone must be reduced. In this process, NAD+ is reduced to NADH.
The 3 carbon compound is break down. 1 carbon is releasing out of the body as carbon dioxide into 2 carbon compound or acetyl-CoA that is attached to coenzyme A.
There is no ATP produced, 2 NADH gain during this step. These 2 NADH will go into the electron transport chain. 2 carbon dioxide molecules are released. For the inputs of the preparatory reaction are 2 pyruvate and 2 NAD+. Outputs are 2 NADH, 2 CO2, and 2 acetyl-CoA.
The main idea of the citric acid cycle or Krebs cycle is the process to oxidize acetyl-CoA and get FADH2 and NADH. The Krebs cycle happens in the matrix of mitochondria.
CoA enter the cycle to produce NADH, FADH2, and ATP. We need 2 rounds of Krebs cycle to break down 1 glucose because only 1 acetyl-CoA can enter the cycle at a time, and we have 2 acetyl-CoA, so we need 2 rounds of Krebs cycle.
Next, I'm going to talk about the step of Krebs cycle. First, acetyl-CoA are 2 carbon compound enter the cycle and attached to a 4 carbon compound. This combination is resulted as a forming of 6 carbon compound. Second step, a 6 carbon compound is oxidized to 5 carbon compound by NAD+, which is electron carrier. A carbon molecule is released out as a form of carbon dioxide. NAD+ is reduced into NADH. This NADH will go directly to the electron transport chain. For the third step is very similar to the step 2, which 5 carbon compound is oxidized into 4 carbon compound by NAD+, and NAD+ is reduced into NADH, and 1 carbon molecule is released as carbon dioxide. So totally 2 carbon dioxide is released in 1 round of Krebs cycle.
4 carbon compound rearranges itself and oxidizing ADP to ATP and rearranges itself again, transferring it to hydrogen atom to FAD, reducing it to FADH2. Furthermore, oxidized by NAD+ to form NADH, became the first 4 carbon compound that attached to acetyl-CoA in step 1.
For ATP, 2 acetyl-CoA give out 2 ATP. For NADH, 2 acetyl-CoA give out 6 NADH, 3 for each. For FADH2, 2 acetyl-CoA give out 2 FADH2, 1 for each. And for carbon dioxide, 2 acetyl-CoA give out 4 carbon dioxide, 2 for each. For ATP, it will be used by the cell process. For NADH and FADH2, it will go to the electron transport chain. And for carbon dioxide, it will go out of mitochondria and out of the body.
Input of the Krebs cycle, 2 4 carbon compound, 2 acetyl-CoA, 6 NAD+, 2 ADP. Output, 4 CO2, 2 ATP, 2 FADH, and 6 NADH.
First step is FADH2 and NADH from the previous process come into electron transport chain. FADH2 and NADH is oxidized, so FADH2 is oxidizes to FAD and NADH oxidizes to NAD+. Both of them leave the hydrogen ions inside of the matrix.
High energy electron of the NADH and FADH2 will release the energy along with passing through the electron transport chain. Those energies will be used to pump proton or H+ from the matrix, which is the phase that has low concentration gradient, to the intermembrane of the mitochondria, which is high concentration gradient. This process creates an electrochemical gradient.
The high energy electron passes through, which releasing energy to form H ion until the end of electron transport chain, where the low energy electron have to leave and goes to the oxygen, which is the last electron acceptor that will combine with the hydrogen ion in the matrix to form water.
Chemiosmosis. First, as we can see that in the intermembrane space now has a large amount of hydrogen ion, which means that in the intermembrane space has the electrochemical gradients produced by the buildup of hydrogen ion, and it is mean that it has large amount of potential energy and ready to be released. Step 2. Hydrogen ion move down their concentration gradients into the matrix. They pass through a channel protein called ATP synthase, and as the hydrogen ion move down to the ATP synthase, the kinetic energy they provide help ATP synthase to add a phosphate to ADP to form ATP.
To know that one NADH can produce 3 ATP and one FADH2 can produce 2 ATP, but this number is just a rounded number, so we got about 32 to 34 ATP in electron transport chain. So for the electron transport chain input, there are 10 NADH, 2 FADH, H ion, ADP, and phosphate. For the output, there are 10 NAD+, 2 FAD+, H2O, and 32 to 34 ATP.
For 1-glucose molecules, glycolysis produces 2 NADH and 2 ATP. Preparatory reaction produces 2 NADH and release 2 carbon dioxide molecules. Krebs cycle produces 6 NADH, 2 ATP, 2 FADH2, and also 4 carbon dioxide is released. The electron transport chain can produce totally 32 to 34 ATP, which one of the NADH can produce about 3 ATP, which means that 10 NADH given from all the steps before can produce up to 30 ATP. And for the FADH2, we get 2 from the Krebs cycle, which it can produce about 2 ATP each. Totally 4 ATP is produced from FADH2, and in this step, there is no carbon release. So for 1-glucose, it can produce about 36 to 38 ATP.
We already learned about aerobic respiration. Next, we will look at anaerobic respiration. Anaerobic respiration occurs in our body when the oxygen is absent. It occurs in cytoplasm of the cell. It also occurs in some prokaryotes that live in low oxygen environment.
Anaerobic respiration or fermentation is the conversion of pyruvate to either carbon dioxide and ethanol or carbon dioxide and lactate. There are 2 types of fermentation. One is alcoholic fermentation. This one is occur in microorganisms such as yeast. This type fermentation produce a lot of food and beverages products such as wine, beer, yogurt, cheese, and bread. The another one is lactic acid fermentation. This one is the one that occurs in human muscle when we are exercise. It also happens to other organisms too.
Lactic acid fermentation. First, 1-glucose enters the cycle, and this glucose is giving 2 phosphate to 2 ADP, forming 2 ATP, and it also oxidizes to 2 AD plus, forming 2 NADH. After that, it results as 2 pyruvic acid. These 2 pyruvic acid are reduced, which NADH are oxidized electron to 2 pyruvic acid. Each of the NADH give 1 electron to one of the pyruvic acid. Due to oxidation, 2 NADH are later become as 2 NAD+, and these 2 NAD+ will go back to the first step. H2 lactic acid is the final product for lactic acid fermentation. Advantages of fermentation. The main benefit of fermentation is the speed of making ATP. It can produce ATP very fast comparing to aerobic respiration. Some of the advantages of fermentation are that it lets your muscle get the energy that need for short burns of intense activities, and it doesn't require oxygen. And for the disadvantages, the lactase and alcohol are toxic to our cell, and the lactate will change the pH and cause muscle fatigue, muscle pain, and cramp. However, aerobic respiration is more efficient than anaerobic respiration because aerobic respiration can produce up to 38 ATP for using one glucose molecule, but anaerobic respiration can produce up to only 2 ATP by using the same amount of glucose.